Blackboard. That's probably a really good idea. I think folks have been finding the recordings useful.
Sorry,
And
this PowerPoint isn't on Blackboard, is it?
it is actually. It's not separately in Blackboard. You have to go to the lecture link. And if that's giving you trouble, let me know, and I can fix that in Blackboard. But for the time being, you can absolutely grab it out of the chat.
Okay, you said we have to go to where?
Do I have a Blackboard instance open? Let me see. I do. So if you go into the course and you go into Blackboard, let me just share my other window. It's this one. And let's see, microbiology, content, and this here links to additional PowerPoints.
Oh, okay.
It should open my drive. And yeah, it's all here. And we're looking for this one up here in the top right.
Okay, thank you.
Sure. Yeah, no, I'm trying to do all right with keeping everything accessible. And I never know where folks are coming from. Sometimes I come in hot from meetings, and that tends to be my Thursdays. So I like to have it on the desktop, drop it around, and share as I can. So hopefully that's been working for folks. All right, so eukaryotic cells and microorganisms. So this is kind of fun. And I don't know why it's fun for me. I remember being in general bio class in 1995 fall, hating this to pieces. And so it's really weird that I find it so interesting today. The nice thing about this is all the stuff you learned in A&P is really going to be useful here, because eukaryotic cells are basically eukaryotic cells are basically eukaryotic cells. And unfortunately, in the lab, we really don't play with too many of these. I don't know why. I guess because the microbes are easier to handle. The prokaryotes are easier to handle. But you do sometimes see aspergillus in the lab. I don't know if you guys have encountered that at all, or if you have seen any of the Saccharomyces cerevisiae in the lab, right? Those would be the representatives from the fungal kingdom. And so I don't know if people do or don't recognize those names, but I'm assuming as you guys get into what should be biochemicals, that some of these tests will come up, and or well, we probably wouldn't show you that, but sometimes you'll see plates with fungi on them, and the borders and the way they look are telltale that it's not a prokaryote. I guess that's one of those things we'll have to describe rather than hope that you see. The cells that we kind of care about here, algal cells, and so we mean protista in this case. And you'll notice the chloroplasts in these cells, the green things in the middle here, are organized very interestingly in the protista that we call algae. There are some called spirogyra, where the chloroplasts are filamentous, and they actually zig and they zag throughout the cell, which is really kind of interesting. In the prokaryotic cells, since algae is kind of a crossover, and I might even use it that way, some of the prokaryotes do some interesting things, so do the protista, because here we are looking at something green, and yet not every photosynthetic pigment is a green one. So chlorophyll A, chlorophyll B, chlorophyll C, they are green, yes. But phycocyanin is blue, phycoerythrin is a hot pink. Some of the diatoms will use, and they are algae, use yellows and browns as their photosynthetic pigments. Some of them use oranges and mahoganies. And I'm telling you now, I don't even know what those look like. You'd have to show me in a Crayola box. But we have names like brown tide and mahogany tide and orange and red tide. And a lot of those are algae. And so we're showing you green, and it's probably one of the biggest lies perpetrated in education, because we expect photosynthetic things to be green. And some of them are, that's not a lie, but the vast majority of them are not. So that's kind of a funky thing. You'll also notice here, you've got green and you've got centrioles, right? Which is also a really funky thing, because centrioles are animal typically, although this is a protista, so fine. But you would never see centrioles in green things, AKA plants. So that's kind of weird. The fungal cells in the middle look very eukaryotic to me, except for the bud scars. And again, it's not fair to say it looks weird to me. Eukaryotes, yeasts, certainly do bud, so they have a method of asexual reproduction, And that's why you see that bud scar. For those who took Gen Bio, you might remember things like cnidarians, like jellyfish, anemones, or hydra. And the hydras and the anemones tend to reproduce by budding frequently. They just don't leave the same kinds of scars. So interestingly enough, you're hearing the word asexual when it comes to eukaryotes, which we regard a lot of the time as sexual beings, but there are some asexual reproduction events that do happen. And then the last one, you see a protozoa, probably not a euglena, not a paramecium, but something flagellated. So this would end up probably in the Mastigophora category, so the flagellates. And again, very eukaryotic appearance. Nucleoli, you've got a nucleus, you've got some Golgi apparati, you've got some vacuoles. So things that you're usually accustomed to seeing, at least if you took A&P and you looked at mammalian cells ad infinitum. So this is a good place to start because we already know the parts. We don't have to beat this up. We don't have to go over this. And instead, we can use this as a survey. And so survey is kind of cool because it implies a bunch of things. One, we're not going to go anywhere near into depth the way we did with the prokaryotes, the way we're going to look at these. With a lot of this, it's more about taxonomy and recognizing the different kinds and how their impacts on the world are versus, all right, let's look at a fungus and let's dig apart their cell walls. Let's go into the steroids in there and the sterols. How are they different? And we can talk about that, but it's less of what we tend to need to do, right? Algae and protozoans are pretty cool. And I love the helminths. I'll tell you right now, if you can, and if you can stomach it, because I know it's this 6 to 9 dinner hour here, Monsters Inside Me is one of the best sites for these little clips. It's, I want to say it's TLC or Discovery Channel related, and it really shows you what happens when these worms get into the body. Some of them are fascinating, like hookworms. If you are walking along a beach or in some areas where there's contaminated water, the hookworms can burrow into your feet. They actually burrow directly in your skin. And then they start to kind of embed themselves and they begin to grow. All right, they're stealing your nutrition. Oh, it's a great show. Great show. And in some parts of the world, this is a positive, if you can imagine that, but it's also a negative. I had friends in Ecuador who openly had on their arms and in other places these hookworms. So you could see them as raised, and it almost looked like a varicose vein, but it definitely was moving. And they would leave them there until they got long enough, and they would just take their machetes or their knives and kind of pluck them out of the skin, wrapping them around the knife or a pencil or whatever it was. Sounds heinous. It is. Sounds gross. Probably. Especially the hookworms that then get into the bloodstream. You cough them up and then swallow them again to finish up their life cycle. So ingenious. But the cool thing about it, or the benefit to humankind, is that the countries and the peoples where you see a lot of these endemic helminths and worms are almost void of autoimmune or arthritis type diseases. The body's too busy fighting the helminth to fight itself. Oh, geez, this would have been the western side of the Andes, but it was not in any city. It was one of these tribal type places. Friends were out there doing bird surveys for an ecology and evolution PhD. And so they moved out there and they lived there for 3 or 4 years while they did their research. So they got to learn about hookworms and how they got rid of them. They bought little motorcycles because they weren't able to drive. Buying groceries was real interesting. They would go to farms, point at the chicken they wanted, and they would kill it and quarter it and bag it for you while it was still warm right there on the spot. So an amazing way to live. But, you know, it's not the US. It's not necessarily the most industrial, especially outside of cities. So if you want, I can try to ask where exactly in Ecuador, but I don't know for sure. But it was a cool project. I'll have to look Phil up and ask him. That'd be a good homework assignment for me. So I guess we'll start with fungi. And it's so funny. In the lab, I have like jars of puffballs and gill fungi. But what you tend to see is there's a macroscopic version of these and there's a microscopic version of these. And I don't just mean the molds and the yeasts, because the things, and this is going to sound really strange, the organisms that make the mushrooms that you recognize, the toadstools or the cups that grow out of the trees also have a microscopic version. And generally, we think of those as hyphae, right? Thin thread-like strands of fungi that pretty much infiltrate the soil and most things that are out there all over. And we only recognize them as fungi when they grow into the toadstool. So there's the asexual piece, which tend to be the hyphae, the stuff you don't see. The mushrooms are the sexual beings of a lot of these fungi. Not all of them, but a lot of them. And so that mushroom, that shiitake or cremini, is actually the sexual product, the baby of the fusion of two of these hyphae that make a zygospore that then make a zygote, and there you go. It's the mushroom that grows out of the ground. So they come in a lot of shapes and sizes. Molds tend to be mainly microscopic, although you do see the macroscopic structures, generally the dark discolorations, but sometimes they're not black, they're not green, and they're not blue. Sometimes they're yellow, sometimes they're pink fungi. So you do see that. And then yeasts, right? Yeasts on your skin, you really don't see. Do you see the macroscopic evidence of yeasts? Absolutely. Certainly, if you had oral thrush or you have a yeast infection, you have seen the evidences of the activity of Candida. And it's not that they don't belong there, by the way. Our bodies are meant to have yeast on it, the right kinds of yeast. They're part of our normal flora, but we don't want them to overgrow, right? And that's where we have the trouble on the superficial layers of the skin. It's this overgrowth that causes a lot of the grief. Oral yeast, the thrushes, oh lordy, definitely an overgrowth, but they're in there anyway. So when you look at the kingdom altogether, 100,000 plus or minus species, who's rounding, who's counting, a lot of them are unicellular or colonial. So they kind of do their own thing or they associate in groups, and few have super specializations, right? They're a little different, right? The ability to bud's kind of cool. Some of these make particular toxins. Some of these can move between tissue types, and that's really where things get scary. Endemically, one of the ones that we're learning about now is C. aureus. And that's the one that started in the city in a lot of these nursing homes and hospitals, and is basically spread throughout New York State. If you Google C. aureus, and I want to see if I can make sure I spell this right, C, that's not A-U-R, maybe it's that. When you Google that, you can find reports from the New York State Health Department in terms of where this fungus is. And they've released the data as of, I want to say, June of last year. And just take my word for it, you don't want to read it, it's everywhere. The complicating factor about a lot of yeast is, all right, they're on you, they overgrow. If you can remove the moisture or treat the fungi, nice statin or something, pretty easy to get rid of usually. C. aureus tends to be multi-drug resistant. And while not everybody is going to die, once you have it, it tends to become a part of your normal flora for life. So down the road, hard to say how that's going to play out. For the people who are susceptible to it and where they can't find a drug combo that works, you tend to see a 90-day decline to expiration. And that's kind of sad, and it's hurtful to think about that. You know, you go to these places to get well and you come back with something that's worse. But this is just the war that we wage. There's always something new, and it probably always existed, but for whatever the reason, may have been transformed or acquired other genes that gave it abilities that it did not have previously. And so you see some strange stuff. You see this pop up from time to time. And I don't know how many times I've mentioned it, but Morbidity and Mortality Weekly at the CDC, which sounds as riveting as it is, has a lot of info, a lot of information on some of these new and emerging critters. And so you can catch data and tabulations that are in there from all the states and some of the territories with some of these emergent diseases. It is kind of cool. When we look at yeasts, we tend to see a couple different morphologies depending upon where you find them, right? So you're basically going to find the classic yeast shape where they're round or ovoid, and they can reproduce very quickly by budding. You'll also find that these yeasts are very resistant, especially in their dried form, to damage, to UV damage, ionizing radiation, They almost act like spores in terms of how hard they are to kill in resting phase. Obviously, if you add water, maybe a little sugar, they bloom, and then they're much easier to take care of there. There was an experiment, true story. Astronauts took the stuff to space and they left it in space, and they also took some to the moon and left it on the moon. And when it was recovered, brought back to Earth, dangerous. I don't know how they got away with this. They found that when they added sugar and water, it still came back to life, even though it had been exposed to ionizing radiation for the better part of a year. So, very robust creatures. The hyphae, and these are kind of cool. They're thin, they're thread-like, filamentous. Um, I'm going to liken them to a spider's web, or a strand of a spider's web. It's silk. Very thin, very hard to see, very translucent. Some of these are, some of these have septa in them, so they're compartmentalized. Some of them do not. You probably would never notice any of this stuff in the dirt if you didn't know there was a toadstool or a mushroom nearby. It's just part of the soil and all the spores and all the goodies that come along with them. Now, the ones that are scary are the dimorphic versions. So stuff like Candida albicans can do this, where they're dimorphic. So you find them in the yeast form, or you find them in the hyphae form, and sometimes they form what are called pseudo-hyphae, so fake hyphae, where it's just the yeast that have budded together to look compartmentalized, and it's not actually a true hyphae. Doesn't really matter. They cause just as much, and they can cause just as much damage in the body. Candida is the big one when it comes to human pathogens. If you want me to take a quick Google here, because that's the one that I'm most familiar with. Let's take a look, see here. Okay, let's see. So let's see how many examples we get. Oh, I really don't want to go there. Actually, here we go. So histoplasma is one. Blastomyces is another one. Jesus, that's a crazy word. I wish I could, I'm just going to share the picture because some of these are crazy. No, yes. So these are some that they're saying are dimorphic and that actually cause infection, aside from Candida albicans. What else is in here? A yeast is a worm. It goes into the same class as Annelida. So they have segments, and they also happen to enjoy human blood. Leeches are interesting because, yeah, they're parasitic. At least they stay on the outside typically. Beer is a crazy attractant. So if you ever want to get some leeches and you have a can of beer open, pour it around, and it's actually an attractant. Leeches, when they bite, secrete some anticoagulants and some analgesics into the wound. This way you don't notice that you've been bitten, or that you're being fed upon, I should say, and you keep bleeding and bleeding and bleeding until the thing is engorged. When we were last on campus, this is the last time I had a leech on me, I was wandering around the property across from Brucker Hall in the United Water property, so Suez property. And I felt something on my leg that did not belong, and it felt cold. Cold? What would this be? And when I looked down, it was a leech. And so I just grabbed a stick or whatever, and I flicked it off. It hadn't had time to do its due. But yeah, Annelida is where they go. So they are segmented. They are true coelomates. They have 3 germ layers, a tube within a tube body plan, and they have a penchant for human blood. Totally true. So there's some good examples of dimorphic yeasts. And, these are threatening. So, and it's got to be somewhere in here, but since it's on the screen and we're thinking about it, what these tend to do, right, is they will colonize an individual first, right? And so colonization, if you want to think of it like in the case of a pimple, where staph can make a pimple, a boil, a carbuncle, it's localized. It really doesn't go anywhere as long as you open it, drain it, treat it, keep it clean, maybe put some antibiotics on it. It's when that boil or that pimple goes and starts expanding into the tissues around it, or it finds a blood vessel and it gets into the blood vessel that you have problems. Same thing with these dimorphic yeasts. Generally, you're colonized first. It's on your skin. Maybe there's some abrasions and it starts to get into the skin, or maybe it just starts to eat, to dissolve, yeah, it's eating you, right? It's decomposing the skin on the surface of your body, and it's making a hole for itself. Now, if it stays in the superficial layers, the epidermis, who cares? But in some folks, the body just isn't keeping the fungus in check. It's growing faster than it otherwise should, and it gets into the bloodstream. It gets into, the places in the body where it shouldn't, and that's where you have the systemic mycosis instead of something just like a tinea. And those obviously are the ones that are really, really dangerous, and folks can succumb very quickly. My mother was an asthmatic, among other things, and she took high levels of prednisone, so 60 milligrams or more for many, 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 many years. And along the way, because of the prednisone, she developed diabetes, just uncontrolled diabetes, which obviously is uncontrolled sugar, but obviously also yeasts love it. My mother was not in the healthiest of form, so I don't know, if I had to guess, maybe 300 pounds, maybe more, I don't know. And obviously that comes with rolls and folds on the body. And between the prednisone, which is immunosuppressing, and the sugar, which is problematic, the day she passed, and I came into the house to help my dad pick her up because she had fallen, all I smelled was yeast in the house, like that fungusy smell. It smelled like a bakery. And I knew there was trouble when I smelled that. And it's a smell I can still smell today, actually. But she was riddled with the stuff, just from the sugar, from the sweating, from the moisture, from the prednisone. And while we really don't know if it played a role in the cause or not, you can be sure it probably did not help. And so when you see a lot of these, or if you're on a floor when you smell this stuff, Mitchum is a really good antiperspirant. And for a lot of folks, especially heavier individuals where there's folds, but let's face it, armpits, under breasts, folds of the buttocks, really good place to put the deodorant so that you don't have yeasts. So it's a kind of an interesting approach, but I can tell you, once it gets into the bloodstream, you have problems, because now they're reproducing faster than the immune system can take care of them. And being eukaryotes, we have to get an antifungal into the bloodstream that has enough oomph to take care of it, and it would work fast enough, which most antifungals can't. The reason for that is these are eukaryotes. We can't go after the ribosome. One of the best tactics here is to go after the plasma membrane, because they use different cholesterols than we do, or different steroids than we do. So instead of cholesterol, you're looking at ergosterol. And the problem is it's slower. So once it's in, it's extremely problematic. Now, if we look at some of these panels here, some of our yeasties, right? Top left A is very, very common appearance of a yeast, even as a colony, or a fungus, even as a colony on your plate. So when you see these, we just call them Ralph. Oh, Ralph's back. You have the fungus. You can see it's filamentous. You can see it's very rhizoid with the margins. It's fuzzy, right? That's just the way it ends up looking. Macroscopically, you'd see this pretty easily. Microscopically, what you're seeing is all of the hyphae that make up this colony, this mound. And in the picture down below, those thin and thread-like filaments are exactly what I mean. Those are the bodies of the organism. And so some of them have septa, so some of them have divisions, like penicillium. Right? Penicillium was the classic experiment and organism that Sir Alexander Fleming used, and by accident discovered compounds that inhibit microbes. So they're septate, they have septa, they're compartmentalized. Many other of the fungi are. However, looking to the right, Rhizopus is one that is not. Why would you know Rhizopus? Oh, mold, bread mold. So rhizopus tends to inhabit the bread in our kitchens, Wonder Bread and raisin bread, those types of things. Ergot tends to inhabit our rye breads. So ergot it's another interesting one, supposedly responsible for the Salem witch trials in the 1600s, where all of the grains, in this case the rye, was contaminated with ergot. Ergot makes the precursors to LSD. And so as people consumed the rye breads and the rye grains, because what else are they going to eat? They started to imbibe the psychoactive compounds from the ergot, and they were more than likely hallucinating. Now, I'm not going to disprove or prove the existence of the supernatural, so not my job. And I kind of like thinking that there's some stuff that happens. Very different than the black bread mold, the stuff that you end up seeing in the bread baskets or in the packages that have sat around a little too much. Sugar tends to be moldy faster. White bread, processed bread, tends to be moldy faster. Ironically, and if you guys want to back me up on this or not, I find potato rolls or potato bread tends to stay around the longest. And for the life of me, I don't know why that is. Maybe it's the starchiness or the kind of starch. Maybe it's how it's processed. Maybe it's just not bleached, so it's more resistant to fungi. Don't know. But I can tell you I can keep potato bread around for months, where the Wonder Bread or the raisin bread is shot within the week. So certainly different metabolic properties there. Here we see some other interesting stuff. We've got in panel A some yeast. In panel B, we've got some yeasts that are budding. So this is kind of cool. Quick asexual reproduction certainly beats the time that it takes to have sex. Although in this case with fungi, sex is kind of an interesting misnomer or nomer. Different species, again, do it a little bit differently. Not all species have sex. That's something interesting we'll mention later. But a lot of the times it's the hyphae that grow together, or in some cases the yeasts that grow together, the pseudohyphae. And you have a plus and a minus strand rather than a male and female. And when the plus and the minus come together, they actually merge the cells and the nuclei, and they form a zygospore. And that zygospore then is what bears the fruit, literally the mushroom of that sexual union. And so kind of a cool process. And it's notably different than roosters, which are like ZW, and humans XY, flies XY. It's plus and minus because of the reproductive differences here in the domain eukarya. So for what that's worth, it's also very interesting. So some other cool stuff. And these are generalities because obviously the temperature ranges here do tend to vary, but they're all heterotrophic. So we'll get that out of the way. There are no photosynthetic or chemoautotrophic fungi. They're all saprotrophs. Yeah, go ahead, Dave.
Yeah, so looking at the last two slides, I know this one says morphology of yeast, and the one after says hyphae. So with this one, what we're looking at would be an example of that.
So that's the example of a picture or a microscope slide of mold. The hyphae, this is really not hyphae. The hyphae is really this. So these are hypha down here.
Okay.
These are hyphae over here. What you're seeing on these slides are yeasts, the pseudohyphae from the yeasts, and then a picture, I guess a cartoonized or a microscopy picture of mold.
All right, got it. Thanks.
No, thank you for asking. Absolutely. And you guys don't have to wait for the chat. If you guys need to unmute your mics, just do it up. Right. So the idea of saprotrophic lifestyles is important here, that they are living off of dead, decaying matter. That's what they're supposed to do. And it's also super duper important to remind folks that like 99.9% of these probably do nothing to humans. Some of them do things to cats, dogs, cows, horses, okay, but not humans. So most of these really aren't pathogenic. Most of these are about a niche in an ecosystem Most of these are about making materials available to the biology that wants to use them. So a lot of the times the decomposers are really recyclers. And I don't know why. I'm kind of digging the whole Chernobyl thing, and I've done a lot of researching on Chernobyl. A lot of folks tell me never to visit. I kind of want to anyway, but apparently it's a little dicey getting down there. But in the ecosystem surrounding the exclusion zone, there's about 30 kilometers of no-go land. And I don't want to say it was arbitrary because I guess it's been effective, but the winds really decided where the hotspots were, not this 30-kilometer imaginary zone. So, for example, Sweden had to throw out milk from reindeer, from sheep and stuff for years and years and years. They had to waste it. And if you lived west of the plant, it was probably worse than east of the plant, although there was an initial burst of southeast radiation. Anyway, in this zone, there is almost a total lack of decomposition happening. And it's worse the closer you get to the Chernobyl plant. It's a little less in Pripyat. It's a little worse. It's a little less still as you get outside of that. But imagine a place where trees fell in 1986 and they still look like the trees that fell in 1986. There's been almost no decomposition from 86 to 2021. That's crazy. And they've demonstrated this by using laundry bags, fill them up with leaves and detritus, and weigh it, and then put it out there and weigh it again. And the mass does not decrease. So when you think about an ecosystem trying to recover, it's got a long way to go. You can't have ecological succession if you can't have decomposition, remineralization, and then new growth. It's a phenomenal disaster. Unfortunately, since we're talking about it, some of these yeasts, some of these fungi are parasitic. They live on the tissues of others, but they don't have to, which is interesting. It's different from our viruses, right? And mycoses and tinea are fairly common. So in general, we think of skin infections, hands, feet, right? Between the fingers, between the toes. And not to be gross, but if anybody has a black light at home, shine the black light over your hands and feet. See if you see things light up. Fungi have an interesting self or autofluorescent property that under the black light, they will actually glow. And so ringworm is one of these. If you use a black light and you suspect ringworm on yourself or on a cat or dog or farm animal, it will glow an eerie green, right? You'll also notice it looks fuzzy. You'll notice that there's no hair growing where this fungus is growing. But the telltale is a spore assay. The easy is a blacklight treatment, or technically a woodman's lamp. Where else? Well, anywhere you have a fold, you could technically have a tinea. So groin, armpits, your hair, your scalp, your chest, under breasts, anywhere there's folds, I suppose the back of the knee, and then in orifices or orify. So the mouth, genital orify, and in nursing mothers, oftentimes the breast, the nipple, or the milk duct tend to be contaminated or overgrown with candida. And mom doesn't generally notice this, although I'm sure there's an odor. The child nursing typically ends up with oral thrush, and that usually is what gives the sign to mom. Growth is typically, and this is the typical part, of the ones that cause disease 20 to 40 degrees Celsius. So they fall smack into the mesophile range. I'm specifying cause disease because I'm sure you've found this stuff that pops up in your refrigerator. All right, pastas tend to go moldy pretty quick. And those types of things where it's at 40 degrees, which is 4 degrees Celsius, but you recognize a fungus when you see one. And so, you got to ask yourself, is my refrigerator functioning? It probably is, but you should have a thermostat in there anyway, a thermometer. Or is this really a fungal psychrophile? And the answer is it's probably a psychrophile, but I would still check the refrigerator temperature. These things live pretty much everywhere. Have not heard of these living in nuclear reactors like our prokaryotes, but they live in really strange stuff that like rubber trees and tires can actually have fungi that degrade it. And when you think about rubber, there's natural rubber, sure. Vulcanized rubber, I don't know about that. But it's thought that this stuff is pretty much everywhere. And it makes a lot of sense. Fungi produce interesting metabolic byproducts. And again, this is going to sound gross, but and I'll show you guys. I've got my Xbox controller down here and I hook it up to the computer sometimes. And I've sat there for hours playing games. And you sweat and you curse and you're kind of locked in a position for a while. And I wouldn't lie if I said, sometimes you can smell like the fungus like starting to grow between your fingers. And I'm not going to lie either. I've hit it with a black light, right? And so a little soap, a little water, and I'm not going to lie about the Windex either. Takes care of the problem. My immune system is kind of weird. I do take Humira, and so sometimes you wonder about what's going to happen. Humira is usually a tumor necrosis alpha inhibitor, and so it's more of a viral problem than it is a fungal or bacterial problem, in case anybody was wondering. At least that's been my experience. I have had, I had a tinea once. It was a good one too. I've had ringworm. That was fun. But I had broken my leg after playing ball. I was playing softball, intramural. Slid, was sweaty, was out in the field, and literally went from the baseball diamond to the hospital to the operating room. And I was laid up probably a good week before I even thought about wanting to get out of bed. And I just remember like rubbing my hands on my body, on my skin, and it had like a film on it or like a coating, kind of like when you go into the shed and you haven't opened it for a season and you start touching stuff. And then the smells and then the discoloration and then the crenated tissue started happening. And I ended up having to get antifungals for about a month. But Lordy, anybody who says superficial has obviously never had one. Now, fungi do have two life cycles. Some of them will have a sporophyte and some of them will have a gametophyte, or really in this case, an N or a 2N, haploid or diploid. Not like plants where they alternate and they spend the same amount of time in both. But they do alternate generations. And a lot of the time, these fungi move around by sporulation, right? So sporangia or sporangia spores, candida or candidophores make candida spores. And so when they're made, they tend to dry out, they peel open, they shoot out. So it's, anyway, I'll show you what these would look like. But it spreads them to the four winds, and then they basically pop everywhere, and they germinate, and they make new fungi. Some will do sex, and I'll show you 3 out of the 4 of these classes of fungi actually rely on sexual reproduction, but not all of them. And so some of these are called imperfect fungi because they are lacking the sexual phase. Now, don't get me wrong. They might have a different lifestyle, but they are every bit as effective as a fungus. And one of those particular species that happens to be an imperfect one is the agent responsible, or at least thought to be responsible, for King Tut's revenge. I don't know if anybody's interested in Egyptology, but this is one of the ways that I like to learn this stuff, is just kind of thinking about the stories and then following up on the science. And so if you're not aware of what happened, they found the tomb, I don't know, 1934, give or take. And yeah, they thought it was a curse, totally. Because many of the pharaoh's tombs were said to be cursed. They wanted grave robbers out. Some of them were booby trapped. So they kind of were cursed, or at least the people who entered them were. So they opened the door, they moved the stone, whatever. And they let air in for the first time in who knows how long, right? 2,000, 3,000 years. The people who did this were obviously breathing very heavily or just breathing in all of the dust and all of the stuff that was in the air that got stirred around for the first time in 3,000 years. And so even though the species that are around today existed then, they hadn't seen the light of day. They had not divided in over 3,000 years. And so they probably were very, very different biochemically than what they are today. And so these folks breathed it in, and many of them died of lung and other respiratory illnesses in the first 6 months to 2 years. Liquefaction of lung tissue, suffocation, hemorrhage, and it was all due to Aspergillus niger, A. niger. which is which ironically is one of the organisms we keep in the lab because it's a great example of a fungus. So it's kind of interesting to think about. They don't have sex. They continually reproduce through budding and asexual means. And so that probably means they have only changed by polymerase error over the last however many years. And it's curious, I'm sure there's studies done of how similar they are from King Tut's time to the time the Egyptologists actually fell ill. So kind of cool. On the left, you see sporangia. So this whole structure is a sporangium. It's one. Sporangia would be multiple. And you see the sporangiospores coming out. Fungi tend to name the spores based on the structure they come from. So sporangiospores come from sporangium. Beneath, you see candida spores come from candidophores. So that's kind of interesting there. And then you also see that not all candida look the same. Arthospores, chlamydospores, phyllospores, macrocandida. Actually, these are kind of cool when you look at ascomyces. What you're seeing here are and will be the remnants of some interesting things. But those are the babies, right? Those are what are going to make new fungi. Porospores. I mean, just kind of cool stuff, but similar but different, right? I mean, they basically do the same job. And so in our, and I love these names, in our fungi, there are four main types here that we care about. The zygomycota, because they make zygospores. Ascomycota, or the cup fungi, because they make ascospores. Basidiomycota, or the club fungi, because they make basidiospores. And then the deuteromycota, right? These are the ones that don't have sexual spores. They do still sporulate, but they're just not of a sexual genesis. They are, or should be, identical to the spores from the parents that produced them in the first place, right? Theoretically, plus or minus some genetic drift through the actions of the polymerases. So you can kind of see why it's interesting, yet a little boring, right? Because it is kind of a list of stuff. So we might as well start with food, right? Whether it's your bread or maybe your cheese, because there's mold in cheese too, why not? The bread mold is not supposed to be there. Sometimes the cheese mold is not supposed to be there. And this is a situation all of us have encountered at some point in time. And I love it because you know what, the heathens that we are, oh, look, it's moldy. I'm just going to rip it off and I'm going to eat it anyway. And yeah, I do that because that's all the bread I have, right? And sometimes you do that and you can just taste the fungus in everything. That makes sense though, because if you see it, if you see these macroscopic structures, it means that the hyphae are already all over the bread and probably all throughout the bread too. And so what you've got happening when you see these discolorations, these black or this blue-green mold, those are the macroscopic structures. And so that tells you that there's already been a colonization. With cheese, I tend to find yellow or more commonly pink or red molds, especially on my mozzarella. A lot of the parmesans tend to be that blue-green mold. Kind of a different situation. I probably would just cut off that. In pizza sauce, you tend to get similar molds. And if you see it in the pizza sauce, sadly, this is what the restaurants do, you can just skim it off. And it's different, right? Tomatoes are acidic. But in the bread, it's already in there. And so you might as well throw out the loaf, unless you honestly don't care. It's probably not good for you. I don't think you're going to die from it. So the hypha are all in there. And eventually, these hypha will grow next to each other. So you've got a little piece of mold over here, a little piece of mold or colony really over here. And so what you'll see is the hypha grow together. And when they grow together, we've got a positive and a negative strand, or mom and dad, if you really want to. And notice they grow toward each other. And when they grow toward each other, it's these two cells that actually merge into one and fuse their nuclei together. So you have a zygote, or really a zygospore, that starts to grow and mature. And when it finally hits maturity, you have this fruiting body that comes out of the zygospore. And this is very common in some of our aggressive fungi in like the rainforests. And so this fungus here is going to bear fruit literally of the sexual activities, and you've got a sporangium. And so the sporangium then will dry out and it will unzip and open and literally fling the spores out of the sporangia onto a substrate. And once they're on the bread, it is very, very easy and fast to see why and how it spreads so quickly. And it's kind of interesting because there are a lot of fungi that do this. There are a lot of fungi that make these fruiting bodies, and then it makes it very easy for it to spread. Let's see. I want to see if I can get a reasonable picture here. Yeah, here we go. Yep, there it is. And so black bread mold isn't something that we're really concerned with. And, you know, you probably shouldn't be concerned with it either. You like it, you don't. It tastes terrible, you throw it away, the end. Interestingly enough, some of these fruiting bodies or some of these strategies are really insidious. And so what you're seeing here is a collection of, well, there's a couple of things going on, but a collection of ants that have had fungal contamination. And the fungus actually grows inside the ant's body, more specifically the brain. And so what you're seeing here is the hyphae and kind of the fuzzy inside of the ant brain or skull. I don't know if I can make that up. There we go. So you can see this ant is 100% infected. And from the brain comes this fruiting body, right? The sporangium. And so what tends to happen is these fungi get into the ant and it controls the behavior of the ant. And what you're seeing is an ant that climbed up on a leaf and bit down on it. And the fungus did not allow the ant to release, right? So it's one of like these situations here. And so the ant literally dies with its mouth parts clamped onto the undersurface of the leaf. And then the fungus does the rest. It fruits and it spreads its sporangia all through the forest. And you see beneath, and I love the art here. I wonder who drew this. You see the ants essentially becoming possessed, right? It's like a body snatcher almost. And you kind of see this again and again and again. It's not just ants that have these problems. Copepods, aquatic organisms, land organisms, a lot of these fungi use very, very similar tactics. And it's really wild. This one has two fruiting bodies. It's really wild when you look at how something that we kind of take for granted, or at least we just say, all right, it's harmless. And yeah, it's makes you think, doesn't it? Where it's kind of harmless and you look at this and you're like, wow, that is super, super eerily similar to what just happens in the rainforest. Now, ants, aliens, I love it. It is a little crazy. It is a little disheartening. Now, it's not just, fungi that have this ability. We see parasitic worms able to exert a control over their hosts also. You see that in cats, and you see it in humans that get infected too. So it is not an uncommon reproductive strategy, especially in the parasite world, to do something like that. And we're just glad we don't live in tropical regions. We just have the poisonous snakes and Lyme disease and West Nile virus and EEE and all that stuff. I wouldn't want to deal with this. No, sir. You'll be happy to know that these don't do that. Ascospores don't do that to ants or humans. And a lot of the time, these are purely ecological. And so you'll notice some words that are kind of interesting. Ascogonium, right? That tells us it's a female. And antheridium tells us it's a male. We borrowed that from plant language. Asci typically means cup. So the ascospores are the spores from inside the cup or the fruiting body. And I do want to zoom in a little bit here. How did the authors do? They did poorly. All right. So what you'll see here is from this union of zygospore, you see new fruiting bodies, or you see the products of sex. And up here, what you typically see are recombinations. So the blue and pink is actually correct. That's what they look like when you zoom in a microscope. But what they're not showing here is the random assortment and the recombinations that you typically see when meiosis occurs. So everything you learned about human meiosis in biology or sexuality actually holds true here. And I'm disappointed that I didn't see this sooner, because the ascospores really should not be the same order in all of them. They really should be randomly mixed up, indicating that they are different from the parents and different from one another because of meiosis. And that's unfortunate that we don't see that here. But everything else is spot on. You've got a male and a female, a plus and a minus. Although here the ascogonium and the antheridium do have different traits. And the end result is the same, right? You have spores then that can colonize and move things along. I've got some amazing, I wonder if I can find them, amazing pictures of some of the fungi from RCC. And I don't even know where to begin to look for these, unfortunately. Because sometimes in the summer you're outside academic one and you kind of look around, you're like, oh, those look really cool. What are those? Oh, those are fungi. That's not right. And they're beautiful, right? They're orange and they're yellow and they're all these colors. They're not going to infect you, but I would certainly caution anybody eating these things. And the challenge there with eating these fungi is they tend to make toxins. They tend to be chock full of poison, actually. And a lot of these toxins or a lot of these poisons are very nefarious because they go after liver and they go after nervous system tissue. And so it's one of these, you definitely don't want to accidentally eat them and pick and guess wrong, because you're taking your life in your hands. And obviously nobody wants to be in that situation when you're just looking for a bite to eat or a meal, and or you're cooking a meal for the family. Oh, let me just go pick some mushrooms. Yeah, that typically doesn't end very well. And I'm just taking a quick look here because I remember about when I saw these. And the irony was I was out there with Dr. McLean, who was like, oh yeah, I know exactly what these are and such, and this, and it was actually a good learning experience, but I don't see, probably because I don't remember the year. I don't see the pictures that I want, so that's a bummer. And I guess it could have been 2020, right? We did start out kind of live. Let me check one more place before I call it a day here. I got nothing. Oh well. Okay. The next one, these do tend to be food sources for us. Obviously, that toadstool looks very, very familiar. And these are kind of cool because of how big they tend to get. So you see your basidium, this is because of its club shape. So in the gills underneath, you have basidia. They produce basidium. The basidium produce basidiospores. So the nomenclature here is pretty boring, but on point. And you definitely have your positive and negative, or your plus and your minus spores, hypha, that grow together to form a zygospore. And these mushrooms are literally the fruit of those unions. And so you've got a stalk, an annulus, the gill, the cap. And it's kind of cool because even if you buy these at the supermarket and they're fresh, you might get shiitakes or creminis. If you take the cap off, set it on a piece of paper, white preferably, let it sit there, cap up, gills down for about 4 hours. Give it a tap and lift it up. You'll actually see a spore imprint on that piece of paper. And it's pretty wild because, yeah, it happens just that fast, and they make spores just that readily. Let's see if I can find a picture of this. Spore. Here we go. This will work. Share. Which picture? This one. All right, so top right, that's actually a spore imprint from a mushroom cap. Or if I look at this one, that's literally what they did by taking apart or taking one of these Basidia mycota, taking off the cap and imprinting it on the paper, right? And so after a few hours, you really get an idea of just how many spores they produce and just how many progeny they really can have after 4 hours of meiosis and just letting these things be wind blown. I don't know, sometimes they're just kind of delicious, right? All right, let's see, what else do we have? So obviously, if we're not thinking about making us sick, although we probably do need to talk about that, we're really looking at decomposers. They're saprobes. They're just recycling for us. They do make some cool antibiotics. Penicillin is a good one from Penicillium. Streptomyces tend to make streptomycin. You do see, let's see, Grisia fulvum makes Grisia fulvin. And that's an antibiotic that we use to, antifungal that we use to make, to treat ringworm. Oh, I'm blanking tonight. Obviously, we use fungi in food. We use truffles in food. Some of them are really expensive. Some of them are really delicious. We make oils and such with them too. But obviously, we're interested more in the diseases. We're interested more in the mycoses, to a lesser extent, the toxin production, because that obviously screws up your foods, and as agents of food spoilage. And as we've demonstrated in history as human beings, look, it makes it look bad, it makes it taste bad, it doesn't necessarily make it inedible. In our world today, you could pass, and you probably should, but certainly going back to the 1600s and beyond, human beings ate moldy food for many, 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 many years. So today we can finally cross that off. I heard somebody say it's not that it's toxic, it just can't be digested. And I just started shaking my head like, you're out of your mind. When we consider mycoses, or the types that we get, right, we can consider them superficial, so very much the outer layer or the outermost layer of skin and hair. And it's kind of interesting, the hair ones tend to produce baldness in large circles, or they grow into these large circles. And sometimes it can be misdiagnosed as a fungus when it's not. But superficial is literally just the outer layers, or even the dead layers of skin and hair. The cutaneous versions are, by definition, more invasive, and they get lower and lower into the epidermis. So there's give or take 5 layers. So they're penetrating 2, 3, 4 layers in. And if you've had invasive or stubborn, is usually the word they use, nail or hair infections, you've had cutaneous ones. Yeast infections tend to be superficial, believe it or not. The other weird thing about these mycoses when it comes to nail fungi is they're mistaken for a lot of other things. They're not always funguses. I happen to have some messed up toenails, and you don't need to see them, but they're the result of psoriasis and psoriatic arthritis. Sometimes autoimmune actually can work on the nails, and it's not fungus, although it looks like it. Fungusy nails tend to be crusty. They tend to be brittle. They tend to be discolored. A lot of the times they're yellow. And it's really strange because the nail itself is dead, which means it's in the tissue of the nail bed. And it's not at the surface of the nail bed. It's usually just underneath. Topicals don't tend to get under the nail. They tend to treat the nail and the tissue around the nail. So a lot of the times you're taking diflucan or systemic antifungals when you deal with this. Many times, and I think someone said earlier, it involves liver tests and it involves blood tests, because you're trying to get out something insidious with things that are quite toxic, and they take their toll on the body after 3 or 6 or 9 or 10 months. Now, sometimes, but wait, there's more. Sometimes the fungi get even deeper, right? Infections of the dermis. So now we're way into the skin. Tissue, you don't want it there, or bone, and that's the worst. Any infection in the bone is very dangerous. Typically, you see lesions, and you are probably going to be at wound care if you don't catch it early, or if you can't catch it early enough and you're diagnosed here. They're going to have to probably debride and continually remove the live and dead tissue surrounding the lesion until you can get it under control. And it becomes a step away from systemic at this point. With Mom's health, I can tell you that she had wounds on her body, legs, where I could probably put about I don't know, $2.50 worth of quarters in there. It was that deep. And we would have to pack it with particular ointments and creams, change the dressings often, and bring her back to wound care often for them to clean, debride, and cut out this fungus and the damaged tissue. Very disconcerting to watch. And I'm sure it wasn't fun for her either. But obviously, once they get into the bloodstream, that's when you have trouble. And they have some structural adaptations. They have some biochemical adaptations, a different gene here, a different gene there, very much in the way prokaryotes would, so that they evade your immune system. And you often see these dimorphic infections in people's lungs. And obviously, lung infections you have to be really careful with, but very aggressive with if you expect to have a good outcome. Now, the part that I love, I really do love pictures. You see some examples of mycoses and some really kind of messed up stuff. So obviously, on the top left, you see mycoses of the groin, mycoses of the buttocks. Bottom left is hair. Middle bottom is feet and toes. Then we see a little bit of ringworm. That's always fun stuff, top right. And you see some nail fungus, bottom right. Right? It's that yellow, that discolorization. And what makes this very different than an autoimmune or a dactylitis or a psoriatic version is you see virtually no reddening or discoloration. It's usually red. It looks angry around the nail. And so it's kind of a clue in that it's not autoimmune or it's not dactylitis related, not diabetic related here either. It's just fungal related. But take a look at these feet and these toes and this butt here. Wow. That's the fungus literally decomposing our skin. And you can see it's particularly worse in the folds. It's interesting to see it in the scalp, and you can see it's fuzzy, and it does produce a very nice round circle. Yeah, that's definitely not pleasant. And it's not so much the ouch. I did have athlete's foot once. I couldn't tell you how it happened, but the itching was phenomenal. And as you itch, it gets under your nails. If you don't wash your hands, it spreads. You use your other foot to itch it. It gets under those nails. So the thing with these is they are transferable. And that's one of the things that we tend not to think about. So if you have athlete's foot and you decide to put your underwear on before you put on your socks, you very easily could give yourself a yeast infection or tinea, a jock itch. You easily could do that. So it's something you wouldn't even think about. Oh, I have a fungal infection on my feet. I should put my socks on first. Makes sense now, perfect sense. Some people don't think that way. My brother was a horrendous child. He used to wear sweatsuits and sweatpants the entire year. Jesus, till he was probably in high school and they beat him up for it. And I just remember my mother telling him she would no longer do the wash for him anymore because his socks and his feet were so yeasty and fungusy that she was getting it from handling the laundry. And so, if you work in a healthcare setting, just think about that. Soiled bed linens, soiled laundry items, socks or underwear from patients that you might just think, hey, I'll fold it, I'll put it in a bag, I'll help you get into the shower. You really need to be careful, wash up and or wear PPE so that you don't make these transfers to your own body. Interestingly enough, you tend to find fungal infections in some places, but not others. Like you find it on the scrotum, you might find them in the folds here, but you would not tend to find it on the penis. And that's just by virtue of it's not in as warm or moist environment as you're seeing where you have the discoloration here. And I was looking at some pictures earlier. Don't ask me why. Last night actually. We were looking at other types of bodily fungi that come up. And why not? We're already grossed out by what we see. And so what you're seeing here, let's find a couple reasonable ones like that, right? You see a discoloration of the skin in and around folds. In particular, breasts tend to be a region where this happens. There was a particularly good picture I had seen looking through here last night where the discolorations were just so. Oops, I didn't mean to do that. Or maybe I did. No. even in males, you can see the same kind of discoloration. This is actually a good color. So this would be the fungus here, here, probably here, all of these spots, here, 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 here. And that's what it actually would look like in humans or on somebody. Oftentimes, it's hard to see there. Oftentimes, it looks like someplace where you might have just been extra sweaty. Nope, that's jock itch. Right, so like that might be or might not be some sun, but definitely you can see underneath on both sides of the breast tissue that there is some discoloration. And so oftentimes you're like, I was sweating. It's hot. I've been working all day. Oh, this is actually exactly the color it would be. But you know, when you show that to a clinician, they're like, I'm sorry, you're colonized. You're full of fungus. I am. Yeah, you are. And that's something that I guess people just live with. But you know, if you ever become autoimmune or compromised that way, it's a problem. Sometimes you see this on the back of the head, and this is obviously under the hairline around the neck, and you can see a lot of it coming down. Your hairdresser, your barber would see a lot of this, right? And it's not just in people my age; it's definitely all ages. Children, especially in their outdoors, I don't want to say catch it, but they become colonized, right? They run through the grasses, roll in the leaves. You're bound to pick something up. And I'm not saying you shouldn't do those things. But, you know, if you start to see these kinds of discolorations, you might ask your physician, hey, what's going on here? You know, I don't remember this before, but it's kind of always been there. I don't remember when it came. Yeah, here's some antifungals. We'll take care of it for you. That's a harsh case. But certainly things that you don't want to see when you're examining a patient or when a patient comes in and they say, I got this weird spot. What is it? Because, you know, then we've got to treat and we've got to deal with it. And I'm not saying you shouldn't, but it does complicate the afternoon just a wee bit. Obviously, we could keep going all day with this. So Candida infections or superficial infections here. Orally, this happens to be vaginally, a cartoon picture if you prefer. The difference here, and you see this a lot on tongues, obviously that's not somebody's tongue, that's the back of their throat. You see the uvula here. This will come off when you scrape it. So if you go to the dentist and you say, oh, my tongue's a little white and furry, he or she will take a tongue depressor and scrape along your tongue. If it doesn't come off, it's probably not Candida. If it comes off, then you're probably going to get an oral rinse to take care of this problem. The one on the left is pretty wild. HIV patients, organ transplant patients, children who are breastfeeding you tend to see that. Obviously, this is just an overgrowth. Antibiotics could do that. A pH that's thrown off in the body can do that. Sex can cause transfers between men and women. So, you know, men aren't immune to this. It's just less seen, and you would probably treat it the same way. So a candidosis is what you're seeing in all of these pictures. The candidemia is when it gets into the blood. And obviously, that's the one that we want to try and avoid. That's the one we don't want to entertain because that's the one that becomes life-threatening. Now, I'm going to hit kind of pause here. I see a lot of oohs. I see a lot of ahs. And I'm certainly not asking for personal stories, but if you work in the healthcare setting, I'm sure this is stuff you've seen. And if you take care, and it's one of those things now that we're doing more of, especially as we grow older, you know, when you handle elder care or parental care, or maybe you're living with your grandparents, perhaps even, these are things that start to come up more and more. And I'm not going to lie, you're kind of oblivious to these things. You know, I watched my toes kind of go bad, and there was really very little to do. My rheumatologist kind of referred me to one place and this and that and the other. My mother wasn't always not healthy. You know, it was just one of these things that kind of happened. And I hadn't been over there in enough time to have the conversation to say, oh, gee, it smells a little like bread in here. What's up? Maybe it would have saved her. I don't know. But my dad lived with her and didn't notice the transformation. If you're in a hospital or if you're in a long-term care facility where HIV or organ transplant patients are, oftentimes when you enter the floor, you smell it, right? You know something's up. People who work with C. diff, you know it. People who work with yeast, they know it too. And so, you know, a lot of this is locker room talk. A lot of this is health club talk or high school locker room kind of thing. College dorm rooms, right? Everybody really needs to wear flip-flops. The military, flip-flops on the feet. The feet are important because that's what happens when you're, you know, in close quarters and things are wet and you share common spaces. Not a death sentence most of the time. It's an inconvenience. You just have to get a good diagnosis and then work on it. And I'm not sure if folks are still kind of around or if they want to talk anything about the fungi. But if not, I'll kind of throw up some other stuff here. And rather than think of something that's, well, it is kind of icky. I guess you guys are right. It is kind of ew. Just looking for my chat window again. Sorry. Where did my chat window go? Hello, there it is. We can think about something that's less terrible. And it's so funny that I say less terrible, but while these algae, protistas, pico-eukaryotes, and some other stuffs, while they really don't inhabit the human body, they do not grow on or in you, they are nuisance items most of the time. So what do I mean by that? All right, well, I grew up near Willow Tree Park, and Willow Tree has this little lake or this little pond in there. And in the spring, it was awesome when the snow would melt and there'd be really few people there and no geese. You could walk around, the water was clear. You could walk around, you could fish. And as the years went by, it was greener and greener and greener. It was thicker and stinkier. And really, it reflected eutrophication. It reflected the additional nutrients from the additional homes in Rockland County over the years. And with extra nutrients, eutrophication is generally nutrients put there by humans, not natural processes. Sometimes they're called anthropogenic. The lake became overgrown or over-inhabited with algae. And you're saying, well, so what? They're going to make more oxygen, and they're gonna you know, make more food for the fish. It's not what actually happens. And what tends to happen is these things, when they grow unchecked, that's really where the problem is. Do one of two things. They choke out the water, they shade the other organisms that need the sun, eelgrass and reeds and those types of things, and they deplete the oxygen from the bottom waters when they die. Right? So you really poison a lake or a stream with this stuff. Some of these things make poisons or they make toxins, and some of them cause paralysis if you consume them. Largely, you're eating fish or shellfish that are contaminated, sometimes by drinking the water, sometimes swallowing some of the water. They cause, let's see, paralytic, neurolytic. So you might have amnesia, although amnesiotic is a different version of shellfish poisoning. Then you've got ciguatera poisoning, Vibrio poisoning. Anyway, they bioaccumulate, and when humans eat these, it causes problems. Beaches are closed, and you see red tides in Florida wrecking things. Brown tides off of Long Island, mahogany tides off Japan. So it's not just a US issue, it's a world issue. Most of these, if not all of these, are considered photosynthetic, but not all of them are green. Kelps, seaweeds, euglenids, pico-eukaryotes, they have a variety of pigments. Some is chlorophyll, some are not. Some are strange compounds that look green and that look blue, or they look brown, but they're not chlorophyll. Some of these are phycobilisomes, so that's your phycoerythrins and such. Some of these are photorhodopsins, so you can even see purples in here. In New York State, we have a big algae problem. And in the Great Lakes up north, you see microcystis being a problem. Microcystis is very toxic to humans. The activity of industry up there is what allows this stuff to grow. And so in addition to microcystis, where you've got poisons involved, you also have waters that are so acidic, they eat metal boats. And so, you know, we have real issues here, and it's a sign or a symptom of the problem. It's reflective of human activity, right? That's really what it is. And as there's more CO2 in the air, there's going to be more plant material. As there's more humans, there's going to be more food for the algae. And so we really do have problems coming up. So, you know, the forms kind of vary a little bit. Some have flagella, some are not motile. Some have cell walls, not all of them do. And so they're kind of curious to look at. They're awesome to study. They're beautiful to look microscopically at them. Let's see, this is the Spirogyra that I mentioned. And I guess you could probably figure out why they're called Spirogyra, because their chloroplasts are literally spiral. And what you see are all of the places where this organism can do photosynthesis. In our euglena, you actually see chloroplasts in them. And this one happens to have a great number of them. Dinoflagellates, these are kind of interesting. Dinoflagellates are the organisms that are responsible for many of the blooms. Atamarince. So Alexandrium tamarince is the red tide organism that hits Florida every year. Not all of the dinos happen to be photosynthetic, but the ones that are, talk about structural adaptations. The ones that are practice something called chlorokleptoplasty. I don't even want to try to spell that. Chlorokleptoplasty. So essentially, the dinoflagellates are kleptomaniacs for chloroplasts. They can actually steal chloroplasts from other organisms so they can enhance their own food production. It's ridiculous. It's fascinating. And then the critters right here in panel C that look just beautiful, shiny, prismy, those are our dinoflagellates. And they look that way, almost as though they're coated with something, are because they're made of silica. So you're really looking at the refraction that glass has, but in the water column. And so these things are heavy. These things reproduce really strangely. They almost do like a pillbox thing, where one side gets bigger and bigger and the other side of the box gets smaller and smaller until they have to sexually reproduce and come back to a reasonable size. Diatoms come in circles, which are called centric. They come in long, skinny ones, which are called pinnate diatoms. And so if anything to stick out here, they make their houses, or there's tests of silica, and they're responsible for about 40% of the world's oxygen, which should say, wow, that's a lot of breathable air. A quick note on the dinoflagellates to the right. Again, not all of them are photosynthetic. And there happens to be one organism in particular that's fascinating. It's called Pfisteria pizzicata. And Pfisteria, as it's called for short, tends to cause massive fish kills. And it'll actually attack the fish, attack the tissue, and cause lesions in the animals, which they then die of secondary infections or toxins. Pfisteria is known to make neurolytic poisons. I remember working with a group and they had a Pfisteria bucket and nobody wanted to open it because you would almost lose the evening sometimes, depending how long and what you were doing in there. If you want to look up on this stuff, it's North Carolina and the Neuse River, where a lot of this was first characterized. The life cycle of dinoflagellates is really weird. It was thought at one point in time to have over 24 different kinds of life cycles. We've since whittled that down to say, no, that's not how they work. But the literature is very interesting. So if you're looking for organisms, or you're thinking about, I wonder what critter I feel like writing about for my paper. Maybe it's a dino, I don't know. Maybe it's a fungus. Maybe you get to include some creepy pictures. That might be fun or make it worthwhile. The classification schemes are kind of cool, and they're very lengthy, and we don't really have to care what all they are. But suffices to say, pigments and cell wall materials are one of the ways we classify these. Food webs, yeah, sure, right? We don't have to beat that up so bad. They produce a lot of O2. They consume a lot of CO2. That's important to note. In areas, especially in the ocean, where you see large amounts of algae, you see heavy amount of CO2 drawdown and sequestration. So if you're trying to figure out how to get rid of all the carbon dioxide in the air, there was a guy in 1991, let's see, 1091, John, I wish I could spell that right. A guy named John Martin, who said, if you give me a tanker of iron, I will give you an ice age. And what he really proposed was seeding the ocean in high nutrient, low chlorophyll areas where there was no iron, so outside of the influence of the Sahara Desert, to bring about a drawdown of CO2 to get us back to where we were pre-industrial revolution. I don't know how we feel about that. I don't know how successful he would have been, but let's face it, if we can't stop burning it, we got to find a place for it. We use algae for cosmetics, certainly. For food, oh yeah, we do. If you guys have had drinks, particularly Odwalla, for example, which Coca-Cola, as I understand, just stopped. They stopped production. It wasn't making them money. Used to make some pretty good product, and you'd always see spirulina or chlorella, or even if you want to think about wheatgrass, right? These kinds of things. And a lot of medicines come from algae, in particular, the coatings of the pills. And so if you go out to the desert in Nevada or Utah or New Mexico, you find these large algae farms that they're growing expressly for the polysaccharides they make. They dry them out in the desert sun, and the gelatin or the polysaccharides from the algae are what make the capsules for a lot of our Tylenol, Advil, Benadryl types of medicines. But of course, you know, with the good, you have the bad, and harmful algal blooms are a real thing. There's a journal, it's actually called HAB, and I don't know if they're up to 7, 8, 10, 12, I don't even know anymore. But there's a local guy on Long Island that publishes it, and a lot of it details the local waters of the Hudson, the Long Island Sound, the Great South Bay, some of the rivers and tributaries, Puget Sound. It is a problem. And let's not forget what happens, or let me ask it differently. The main profession back in the day of Long Islanders were fishermen. And in particular, they were baymen. And you wonder, or you ask yourself, what the hell were they fishing? Well, the answer was clams, oysters, and scallops. And if you go down to Jones Beach, you can still get clams today, not the quality or the kind that you used to get, but the scallop is virtually gone from Long Island. It was a 3-day season last year because of how polluted and how many have died off. And you have to think about the harmful algal blooms on Long Island. When you have oysters and scallops beneath, they're filter feeders. So they're siphoning and filtering all of these dinoflagellates and harmful algae and accumulating the toxins. Well, guess what? Some of them are dying themselves. And so in Billy Joel's, or in some of Billy Joel's songs, with Downeaster Alexa, he made the claim you can't be a bayman anymore. And he's right. You know, that industry has died because of pollution. And I'll tell you what the cause here is. It's an imbalance between dissolved organic and dissolved inorganic nitrogen. So essentially, it's all the sewage that goes into these bodies, especially when the rain lets up and it doesn't dilute it anymore in November, December, and January, that you see the most damage. And that's disheartening, that we've broken the ecosystem to the point where, you know, forget people's livelihoods, although you can't, it's important. That is a source of damage here. Forget the beach closures, forget the monitoring costs. The ecological damage will never be undone. Would you believe the Hudson River used to have oysters? You probably can't believe that. The Chesapeake Bay used to have oysters. And they're the first to go when the water quality goes to hell. Right? So let's just think about food webs and how this affects everything. Interestingly enough, the microbial communities love harmful algal blooms because they can be digested by the bacteria. Unfortunately, when you digest harmful algal blooms, you remove all of the oxygen from the water. And so the bacteria might be happy, nothing else is. And this is why you see levels of oxygen in the water go anoxic, so less than 0, hypoxic, less than 2 milligrams per mil, from a normal 6 to 8 milligrams per mil in the water column. So kind of disheartening, kind of scary when you actually sit there and look at it. And I'm looking in the chat. Nobody seems to want to add anything about algae. That's cool, not a problem. Maybe folks care about protozoans, right? And obviously protozoans do cause disease, giardia, amoeba, certainly two that we think about all the time. And there's some other interesting ones. There's probably some flagellates that we care about, but largely we're looking at less species than fungi, believe it or not. And you're looking at about 65,000 plus or minus species of protozoans. Most of them kind of live alone, although you do see colonies of protozoans from time to time. Most of them will swim. They'll have some sort of locomotor structures, usually flagella or cilia, but in the case of our amoeba, it's going to be pseudopods. They tend to vary quite a bit in shape, actually. Most of these don't have cell walls. And most of these don't have chloroplasts. Most, a few of them actually do. And when you look at these, sometimes you see them in a resting stage, so a cyst, or the trophozoite stage, which is a motile feeding stage. And even a third stage, those trophozoites can turn into swarmer cells. Basically, they feed ravenously, they reproduce very quickly in response to unfavorable environmental acts. So basically, they know they're going to die. This is their last-ditch effort to try and save themselves before that's the end. And let's see, I just want to show you, because people ask often, what's the deal with some of these things, or what's the deal with these amoeba that people have? And so the brain-eating amoeba doesn't look anything crazy, right? They just look like regular amoeba here, although that looks a little scarier. I think somebody's playing games with some of these, but these are electron micrographs of the actual amoeba. And what tends to happen is folks who are inoculated, who are seriously unlucky enough to end up with one of these infections, is usually from a swimming pool that has been improperly chlorinated, or a lake or river, fresh water, in a temperate region. So you're thinking Texas, you're thinking Southern California, although I don't think it's been seen there. The South, the American South generally, and Southeast is where you see this, Georgia and Alabama and such. And the water droplets contain the organism. They get into your nose and they get up just a little bit too far. And they start to attack the frontal lobe and the brain, right? So they're literally trying to eat and digest the brain. And what you end up seeing is a swelling of the brain, which then causes pressure to build up. And folks who contract these amoeba generally don't survive. I think out of the 500 cases, we probably should Google how many. Out of the cases of all of these, I want to say one person has survived ever. So let's see, 97% fatality rate. And yes, and the people who survive are not all there anymore. 2016, that one I've heard of. Yeah, so you start reading this. This is the CDC site. And it was something about after 35, this is ridiculous, after 35 years without a survivor, right? In 2013, 2 kids actually lived. 12-year-old girl who was diagnosed 30 hours after, someone else 36 hours. They gave her some sort of new medications, and these particular girls made full recoveries. Well, at least the first girl did. This male did not. So it seems now we have some better strategies, but most of these people do not do well. And that is sad. You're right. We don't see it in the north just because of the chlorine and the climate and our winters. But rest assured, it's probably going to start moving up the coast like everything else. So it's just kind of one of those, swim in areas that are tested. State parks are usually good, but bad for other reasons. And make sure your swimming pools or hot tubs are properly chlorinated or brominated. All right, active feeding stage of a protozoan kind of goes to a cyst phase. Looks familiar, right? Yeast, spores, unfavorable conditions. And once moisture returns, so this could even be a vernal pool, right? Seasonal monsoons could cause this. Once the rains come back and the cell begins to absorb moisture, we go back to this active feeding metabolic stage. So kind of cool. In this phase, you're not going to destroy them. In this phase, it's going to be really hard to kill. But again, most of these are ecological niches. They're not actively looking to harm people. With relatively few cases, the amoeba, Giardia, and a couple others. They're all heterotrophic, so that kind of makes sense. You see a lot of these in moist habitats. They have a variety of sexual production here and asexual reproduction. Some are parasites, and I love when I see some can be spread by insect vectors. Why not, right? Like, certainly the fly and the mosquito have been amazing perpetrators in spreading or acting as viral or arthropod vectors for a lot of things. Viruses, certainly, but protozoans, yeah. And what they end up doing is help act as part of the life cycle. So part of the life cycle is in the arthropod, part of the life cycle is in an animal. And if you don't have both parts, you don't end up completing the life cycle. So that's actually a thing. I'm just trying to think about some of these. So one of the best ones that I could probably give you an example of would be like malaria. And the CDC has a pretty good picture of what goes on and how it happens and what happens and all that good stuff. But it's definitely not for the faint of heart there. Anyway, you control mosquitoes, you control a lot of disease, I guess, is what we're usually saying. There's 4 general classes. So flagellates, amoebas, ciliates, and things that we call amphicomplexa. Now, of course, we can't just call them flagellates. We have to call them mastigophora. You can't call them amoebas. They're called sarcodinia. Ciliates are ciliophora. And the ciliates are some of the most interesting of these organisms because of the way they transcribe and translate DNA. So when we looked at our microbial DNA and how we do transcription and translation, they were unwound, they were unzipped, they were linearized, they were complemented or copied, and then zipped back up. Cilia don't bother with the unzipping. So their genomes are usually circular, but they're more spiral kept. And instead of linearizing them, they go straight through the spiral and back and forth through the spiral to do their transcription and translation. So they're actually jumping across many strands of the genome multiple times to get all of the DNA turned into RNA and all the RNA turned into proteins. It is one of the strangest things I've ever seen, but it's reproducible. You can read about it in papers. It's actually really, really slick to look at. But these are the 4 classes. And I don't think we're seeing anything particularly unusual. Certainly flagellates look like flagellates. Amoebas look like amoebas, except that one on the right. I guess we typically show the amoeba on the left, or that sort of pseudopod. In reality, though, who's to say they can't be long and pointy or other oddly shaped? And when you do surveys of these microorganisms, you actually find some really strange stuff. And if you were so inclined, I don't think anybody is, you could order these from Ward's Scientific or VWR and have them shipped to your home and look at them and study them. Most of these that you're going to buy are not terribly dangerous. And with a reasonable microscope or hand lens and a slide, you can do the investigations at home. So flagellates are probably the fastest moving, if I had to say. The amoebas, pretty slow with the pseudopods. And probably our cilia come in right in between. So this is your typical paramecia, contractile vacuole and food vacuoles, macro and micronucleus, kind of an interesting adaptation here. And all we're showing you is that where flagella work in a 360-degree fashion, where 100% of the motion goes to really pushing the cell forward. Here, we've got about a 50% conversion of energy and motion. So the power stroke pushes you forward. The recovery stroke is when the cilia reset and the organism kind of glides or spirals through the medium rather than actually being pushed forward. Then we've got these. Exactly like a rowboat, exactly. Best way to describe it. Power stroke, recovery, right? Recovery, power, absolutely just like a rowboat. These are weird. Amphicomplexa, all right? It's not clear their method of locomotion. It's not exactly clear what their niche is, but clearly they feed. It looks like they bring in vacuoles through endo or pinocytosis. They have some interesting life cycles. Very poorly studied, though. And so if you're interested in looking at some of these, make sure that there is enough information before you go forward and you say, aha, I want to study Amphicomplexa, because you just might not have enough material. Right, and as we kind of go on, and actually we're doing pretty good tonight, we end up here in helminths, because these are the most fun, and we save the kind of creepiest for last, and they are kind of creepy. So here, these are not protozoans. These aren't oh, what do we call it? These aren't prokaryotes. These are legitimate, actual kingdom Animalia organisms that are getting inside the hosts, right? So literally a monster or an animal inside me. They are every bit as much as an animal as you or me. Granted, they are lower on the scale, so that is important to say. The phylums here are typically platyhelminths and or ashyhelminths, or you might see them as platyhelminths and nematodes. So you're looking at flatworms and you're looking at roundworms. They are multicellular, absolutely. They're not all coelomates. So the platyhelminths and the nematoda certainly don't have any of the fluid-filled line cavities with mesoderm, but certainly there are higher worms that fit into this. Parasites, so they're stealing your nutrition more than anything else, and they do this in a variety of ways. Most of the time, we're concerned with attachment. So tapeworms have a scolex, and they burrow in to your tissues so you can't get rid of them. Nematodes burrow in with their tails into rectal or anal tissue so that they can't be shook loose. And a lot of the treatments for these organisms come at drugs that dissolve the point of attachment. In some cases, we just give you a straight up poison. If you've got cats or dogs, you know when you get a kitten or a puppy or a cat or whatever, you have to deworm them. And that's pretty true. At some point, they're commensal, but when they stick around too long, they do damage to the cat. They do damage to the dog. They do damage to the human. You become malnourished. You might become anemic, and they spread. And that's when you have dangerous infections. There have been stories where tapeworms eat the organs, the abdominal organs of humans, right? It was in there. I didn't think anything of it. And then it killed you, right? So problematic right there. The other problematic piece is that these things are lean, mean, reproducing machines. So if you look at a roundworm, a flatworm, they are filled to the gill with intestines, vas deferences, spermatic tubules, ova, and eggs, right? Yolk glands, things like that. Anything that you would need to reproduce. Seminal vesicles, right? So that they can transfer sperm from one another. Unbelievable, right? And even the fertilized eggs can cause disease. And so, not trying to like scare folks or anything, but there are a numerous amount of these things that even exist in New York. I can't tell you how many years I fished. We had tapeworms all the time. I had a friend, Mark, and you could just hear, hey, like Peter Griffin. And next thing you know, you get hit with a tapeworm. And it was constant. It was unbelievable. In the tapeworms, the flatworms, which are pretty wild, they're segmented. And the segments are called proglottids. I'll show you some pictures. The proglottids contain hundreds or more fertilized eggs. And each of those segments or proglottids can cause and create new tapeworms. So you want to be super careful with that. We got trematodes and flukes. So flat and non-segmented worms, again, sucking mouth parts. Roundworms are nematodes. Things like Ascaris fall under here. And obviously you've got hookworms. Hookworms too fall into this category. Those are the ones that aren't necessarily swallowed, but they end up burrowing into your skin, into your blood vessels. These are the ones that you tend to cough up in the lung and swallow. Again, unbelievable reproductive systems. They just want to make more of themselves. Ultimately, killing you is not their goal because you're their meal ticket. They don't want you to go away. We generally have to care about 50 different kinds of these. It doesn't sound like a lot. It's a lot. And a lot of it is contaminated products. So contaminated water or food that's been contaminated by washing in bad water, or a lot of the times people who don't wash their hands before they handle food products. Now, in fairness, some of these come from insect vectors. In Texas and in Oregon recently, and California too, there were cases of river blindness where people got flies in their eyes. The larvae were in the flies. The larvae then were in the eyes, and they experienced some blindness. The eye swells, it causes massive damage, and you have to act very quickly to save your vision. Totally reversible if caught early. Some folks don't have 20/20 vision anymore, but you're just very happy to see. Other people have described these helminths getting into the eyeball itself, into the vitreous and aqueous fluids, and they'll actually describe seeing the worm swim in front of their iris or pupil, and they actually see the worm. Not so good. Some folks end up with nematodes in the legs or the testicles or the ovaries, and generally you see cases of elephantitis. Some folks, by swimming in contaminated water, can actually have these things burrow into their skin. There's something called a botfly. I don't know if anybody's heard of a botfly. That's a good video. Maybe we'll watch that one. Botflies lay their larvae in the skin of humans, and they actually grow in your skin. There's a hole at the surface that they breathe through. And if you leave them be, obviously they continue to eat before the flies emerge. Again, I don't know about you, but I don't feel like being parasitized for flies to make a living off of me. Definitely a very skeezy kind of thing, but nevertheless affects billions of humans. So these are very pervasive and they are pretty common. And that's like the tip of the iceberg, right? So, here's trypanosomes. You have a bug that you get from an animal, a cat. Basically, then it's kind of cool. It kind of bites the face. The parasite ends up in you, and then the bug basically goes on its way. So it can affect humans, although it would prefer to affect other types of bugs. One of the more interesting ones that's coming up the coast is called the kissing bug. I don't know if anybody has seen that one, maybe. That's typically what's exposed, and that's typically where they bite. So these critters here, right, sometimes are called, or sometimes caused Chagas disease, or sometimes these are the critters that I mean. Where are we? All right, so take a look at that critter. Kind of looks a little like a cucaracha, but it's not. They fly. And they're called kissing bugs because they bite the face. They probably bite the face because it's exposed, or they bite the face because the capillaries are there, and it's an easy way to catch a meal and infect the host. There have been, and that's like kind of a big bug, right? There have been cases, you see Ohio State, you'll see cases of this in Delaware, and I believe there was a case of this in New Jersey. Right, there's an environmental pest management, 614 is Ohio. So yeah, this is not just the Southern United States. This is a real thing. I figured I'd get a lot more ews. I guess you guys do a lot more work and see a lot more ridiculous stuffs. Right, the oral route, very, very common. So you get cysts in water or food. They make it through the stomach. That's where they return to their feeding state. They become adults in your intestines. They return to their feeding state and you pass it in feces. Then it enters the water and it enters the food sources, just primarily through breaks and improper sanitary conditions. And it repeats again. Largely, you see loss of weight here. Excuse me. And sometimes up to 10 or 12% of your body mass. A lot of the times you do experience diarrhea, watery or greasy stools with these kinds of infections. And so you would know that there was a problem before it was diagnosed in some other way. I hate to say this, but I'll volunteer it. Might as well not be the only one who's embarrassed tonight. We found and we took in some cats. And one of them is just kind of a jerk. She likes to be fed. She'll tolerate pets if one of her brothers is around, but just a terrible animal otherwise. And because of that, she didn't come near us when we brought her in. We were unable to deworm her properly. And so before I knew it was the water system malfunctioning in my neighborhood, I thought maybe I had contracted some worms from my cat. And so I actually had to go and provide a sample for the doctors then to go and look for the cysts in the stool, just to rule out the fact that I may or may not have any worms. So they're not looking for adults, but they're looking for eggs and cysts. Fortunately or unfortunately, I came out, yeah, totally fun times. Although I will tell you the true part of the end of the story, I might have provided the sample, but I wasn't the one who dropped them off. And I know that sounds horrible, but I definitely put one of those little Panera handle bags with the samples, with the absorbent stuff in there, and my wife got to drop them off for me. So yeah, right, kind of that spousal love. The things we do for our partners. That's a tapeworm. I'm sure nobody wants to deal with that. Tapeworms are flatworms, so when you look inside them, that's all they do. They eat and they reproduce, and they eat and they reproduce some more. And what you're seeing, all of these segments, 1, 2, 3, 4, I don't even want to count them. These are all proglottids. And what you end up seeing is the fertilized eggs inside. So this can break off, and this can bring with it hundreds and hundreds of eggs. This is the scolex, I should say. You've got suckers and some hooks. Yeah, it does look a lot like a vesicle that comes off of a presynaptic neuron. Absolutely, synaptic vesicle, there we go. And the scolex here is what keeps this growing tapeworm inside your body. It's what prevents you from passing it. And so some of these can be meters or multiple meters in length. One of our faculty at Westchester, hopefully you don't know Dr. Cook. God, it's been so long. Her husband contracted a tapeworm, and this was like in the 70s, early 80s. And she, being the biology and the microbiologist she was, took him to the doctor, took his medicine. The medicine dissolves the scolex, so the suckers kind of don't work, the hooks go away, and he started to pass it. And she literally went in there into the bathroom, grabbed the pencil, and started winding the worm around the pencil. I think partially to make sure she got it all, but partially to put in a jar and show in the lab. Right? So she totally took a sample from her husband and made it into a scientific thing, which, I mean, that's what scientists did back in those days, right? Yeah, I might have shared that one. It's kind of crazy to think about that's what people do. I can tell you that we have at RCC, from older faculty retiring, a very morbid collection of things that's probably only rivaled by the modern Museum of Medical Oddities. Brains in jars, and I've put my fair share in there. Kidneys and hearts. We also have a couple fetuses that were donated in jars already. I'm assuming you mean while they're biting. And if they're biting you, you probably don't want to squish or squash them because they would be likely to push more parasite into you. You would probably want to try to get them off, or at least squeeze the site around where they're biting in hopes that then nothing gets pushed in and that you can pull them off. I probably would not try to kill or squeeze it while it were attached. I think that is a fair thing to say. I also probably wouldn't grab a cigarette and burn it either. Much like a tick, that's probably when you get more of the parasite rather than less. And of course, the last little critter here is our pinworm. And the pinworm looks a lot like just a general Ascaris, where you have males and females who do the do in your intestines. And then the female will come out at night while you're sleeping, generally a child, but it could be mom and dad. And while the child is sleeping, the worm burrows into the kid's anus and lays the eggs. And so the characteristic behavior is hands in the pants and itching because of how aggravated the tissue is, how itchy, how raw, how just broken up it is. The crazy thing about this is these eggs are highly infectious. They are very easy to pass around. And so again, another embarrassing story as I find some pictures for us. My sister and all of us really were, we grew up in Rockland. We grew up in the East Ramapo School District. We went to East Ramapo schools. We went to public schools. And my sister in kindergarten, so this would have been 1991, had some sort of issue. They were taking her to the pediatrician. And I don't know how old I was, 8th grade maybe. So we were doing renovation on the house. I had a Nintendo. It was hooked up in the living room. We were going to town, and I just see my dad take my sister. They're out. Take my sister. They're back. All of her linens. They're out. Vacuum cleaner in the house. Vacuum bag out of the house. What the hell is going on? And it was like 2 nights later, after the vacuuming and the cleaning and the bed linens got changed, and she got new underwear to keep the eggs in her pants. They were like, okay, your sister had a pinworm, and it's not a big deal. Take one and pass it. And we all got this card with poison in it. Passed it around to the table at the dinner table. All right, I'm taking mine. Everybody take theirs. And literally, talk about treating the family as a whole. Took the poison to kill the worm before we were all infested. And so it's pretty wild to think about, 1990 America, 2010, 2020, part 2 America. This stuff is actually happening and it's actually real. And so let me see. This is always a good time to find some pictures and see just how much dinner folks can keep in. And while it's not my goal, it is kind of fun to look at this stuff. So let's see. Pinworms are pretty easy. That's literally what they look like. So if you were so inclined, you could check for these. They are 2 to 5, 2 to 6 millimeters in length, but they are very small. They are very translucent. They will be moving if you find them. And it's kind of crazy. Like as a close-up, it might look something like that. Obviously, we don't want to think about these things in our bodies, but typically 2, 3, 4, 5-year-olds, it's very common. These eggs are in the grass. Kids pass this stuff around all the time. It's really not a big deal. You just have to deal with it. And you start to look and you kind of wonder what the intestines all look like. And just the way you think, right? Worms, worms, worms. The bigger version of this is the Ascaris. And this is the one that we typically show because it's easier to dissect and it's easier to look at. And when you dissect these things, which is what that looks like here, all of that on the inside is reproductive structures, new worms, and its intestines. So again, extremely lean, mean reproducing machines. And I'm sure we could find something terrible if we wanted to. I talk about things in jars. There you go. Dr. Nick, yeah, that's probably perfect. I don't want to think about that. Google, I must have safe search on or something. There's really nothing to, wait. Yeah, no, I don't want to hide anything. Turn off. Turn on. Oh, good, it is already off. Okay. All right. Certainly your idea of classical elephantitis. And when you have nematodes in your body like this, the scary thing about it is those aren't your legs anymore. Yeah, there totally is. Safe search keeps it clean or keeps it G depending where you are. So some of these like are really bad, right? And in endemic regions, exactly right. And so they're literally siphoning off your body, your sweet, sweet juices and nutrition, and they're making more body mass. The classic one is like elephantitis of the scrotum, where literally your testicles and your scrotum are now filled with worms instead of you. This is also true in ovaries, maybe less so in humans, but certainly in fish. It is amazing to see the amount of worms you pull out of fish ovaries when you do the dissections. Quite frightening. And it really doesn't matter where you are. Once they get in, they do some hella damage. These get particularly fun. Sure. Right, where you can actually see the worm inside the eye, or multiple worms inside the eye, or multiple worms inside the eye. Right? These are real pictures, and these are frightening pictures to think about it happening to us. Right? You can see the worm here really clearly in the white of the eye. And these are really damaging. These cause ocular inflammation. These will cause blindness. And so you don't want to risk anything other than a trip to the doctor's visit to work on getting that out. Yeah, oh, 100%. 100% horror movie fuel. Absolutely. Well, you want to know something. Even getting a mosquito in the eye is really damaging because, oh, let's not use this. Let's not use my personal account. We don't want to see that. I'm going to use this one. I'm going to use that one. Where'd my screen go? Share. I think that's what I want. That is what I want. And we want to see. Let's see. That one's much newer. That one's, well, these are all newer than the ones I like. Oh, this is the one I usually watch. So here's another example of a worm. This is actually a fly larva that affects humans. So the fly bites and injects the larva into the body, and then the body harbors the developing larva. And just wait till you see the hole. There you go. So I know that could be better in terms of what it looks like, but notice you see a hole here, and let me just back up a little bit.
Why did they duct tape it so it can't get oxygen?
Yes, exactly right. They're trying to suffocate it. So some people use duct tape. Other people would prefer to use Vaseline. It's really just a taste and preference thing.
They're not like they say duct tape fixes everything.
If it moves and it shouldn't, use duct tape. That's what I say.
Oh shit, they're big. Oh, okay.
So they're going to try and get these out of this guy. And this is Panama. This is not the United States. So.
Oh my God.
Don't throw yet, because I want to see it when you get it.
I can feel it trying to pull back in, girls. You mean it's still alive? Keep hold on, baby.
Keep it firm.
I don't want to rip it though. Yeah, ripping it would be disaster.
And it's fighting back, right? It's trying to stay in there.
Only because we're biologists, we can actually stand.
Oh my God,
Right.
it keeps coming.
And you heard her say, just because we're biologists, we can actually stand it.
By yourself, Kyle?
This is fine, yeah.
Oh,
They are huge. How long does it take to?
Oh, yeah.
Put it on something. Put it on something. I want to...
Ah, it'll be all right. Give it an hour.
Right there, right there, right there, right there.
So look at the size of that fly larva.
Okay, I'm gonna wipe it with that.
There's
And
another one. Holy shit.
Yeah.
Real.
You want me to still alive?
Put a quarter by that thing.
Yeah, definitely put a quarter next to that.
Approximate size.
No, there's another one here. Can I wipe it with this one?
All right, near a shotgun shell, whatever.
Can you feel that?
So that's clearly a very, very large larva. No, you don't want to see zits. Oh, that might be interesting. Let's see what they're doing here. That's too loud. Let's see. They grab it. God, I still don't even have this thing open yet. Holy smokes, there it comes. And I've seen these removed from necks. I've seen these removed from heads. They're going to give this one a kind of a little encouragement to try and get it to come out.
What did you do for a living that you've seen this come out of somebody's neck?
Oh, me? No, nothing. I'm just a biologist that watches gory stuff.
Oh, geez.
I've traveled enough, but I've never actually been places where we've done this.
I had a slug in my hair when I was a little kid and nobody could find it. Like I went to the nurse 3 or 4 times, just like telling them to get it out. And this is my worst nightmare. My
Having
worst.
something like this stay in there.
Yeah.
Oh yeah.
Anytime I feel anything, I don't know what it is. I'm not okay with it anymore.
Well, I mean, that's kind of understandable, right? I mean, nobody wants to have this kind of mess going on.
I'm thankful to live in the United States.
This is another creepy one. So this guy got infected from swimming in water that had nematodes in it. Oh, the background music's great.
This memory. So a friend of mine just got back from holiday, and they're like, Harley.
Yeah, let's see what happens.
Or whatever. So basically, they just squashed a cockroach on this guy's face. This is crushed up cockroach that is smeared in this guy's face. They're telling us some herbal tincture or some bullshit. It's actually a squashed roach. This is a little trick they do in certain little clinics. And now it's called entomopathogenic nematodes, which are little worms, little maggots, which appear to be coming out of the guy's face. If you can see closely, you'll see a little wiggly, there you go, just there, a little wiggly worm. But all it is, it's the nematodes that lived in the dead roach, the parasitic worms from the cockroach, the apparent healer squashed on this guy's face. This should be a bit closer here. You have to see him.
Check out his eyebrow.
There. So the white squiggle on his eyebrow. So it's just a total scam, man. But people gullible enough to believe in it is just.
That's pretty wild. Definitely not watching House MD. Oh, what's going on here?
From a cunning parasite to some of the most shocking cases involving it. This is what happens when tapeworms attack. Before we begin, be sure to subscribe to They Will Kill You. Hit the like button and request any topics you'd like to learn about in the comments section below.
I might have to do that.
The tapeworm, or cestoda, is a parasitic, segmented flatworm, known for invading vertebrate animals and living inside their intestines. They can also infest the human body, and several species are known to cause diseases. Tapeworms don't have mouths and get their nutrients directly from the host's gut. They are hermaphrodites, and each segment of their bodies, called proglotids, contains a complete
Boring.
of eggs in Mexico. After the eggs hatched, her daughter's intestine became.
Oh well. I guess that's a sign. Anyhow, right, the helminths, really interesting creatures, certainly inhabit lots of interesting ecosystems and habitats. You just don't want them inhabiting you. So a good bit of advice is if you're going to travel, keep your shoes on the beach, wear some sandals, try to stay out of contaminated water, and obviously don't eat undercooked or raw food. Doing all the fishery work that I used to do, tapeworms we used to see all the time. And it's terribly common in fish to see tapeworms. And it's really kind of freaky when you look at what tapeworms in fish look like. Sometimes you can see them moving around if you get fresh fish. Right, you can see them actually in the fish here. That would be salmon with a worm in it. But a lot of the times you'll see them when you go to fillet them. Is that a good one? And so you see this worm here happens to be a tapeworm that's in the fillet of the actual fish. And if it's fresh, you can pull it out, and you probably should. However, sometimes you can actually, or sometimes you don't actually see it because it's in the flesh itself, and you cook it. And I remember sitting around one night at a fish fry where Laura was having a nice sandwich of flounder, I want to say. And she screams out, "Keith, you jerk, you forgot to take the vein out." And we're all looking at each other like, "Vein? What?" And we let it go. We didn't want to say anything. But basically, Laura was eating a tapeworm. And since it had been cooked and it had been heated, there was no risk to herself of contracting a tapeworm. But all of us were just like, better her than us. So in particular, fish ends up being probably one of the more dangerous things because we tend to have sushi, we tend to eat it raw, and that tends to be where we're exposed. And a lot of the fish worms don't end up in intestines. Unfortunately, they end up in brains. So be careful with that. And of course, I would be remiss if I didn't mention trichinids. And I kind of did leave those out. And those would be things that end up causing trichinosis. And let me just throw this on real quick. Basically, you're told never to eat undercooked pork or more recently bear because of trichinosis. And what you end up seeing are the worms themselves. They look like this or they look like that. And they end up getting into the muscle tissue where they form these cysts. And so it's incredibly painful. It's incredibly hard to get rid of. And yeah, you could die from this kind of condition, right? And that's literally the worm in a cyst in the muscle tissue. In America, believe it or not, since 1956, there has not been a single case of trichinosis driven by pork. All of the cases we've seen in America have been driven by bear and the hunting of the bears. And it's actually pretty wild because a year ago or 2 years ago, the FDA revised its cooking guidelines for pork. And unlike chicken, you actually can cook pork to a medium rare now. And I don't like it that way. I don't like the way it tastes that way. But what they're really saying is you don't have to kill it. It doesn't have to be killed twice. It doesn't have to be burnt to a crisp. You could eat it mostly cooked if you want to. Who's eating bear? Well, so bears are hunted in New York. Bears are hunted in New Jersey. So they're a great source of meat. They're a good source of pelts, coats, fur, that kind of stuff. I don't know how many people. I've had bear meat. It's not bad. But you just have to make sure you cook it thoroughly. You need 180 degrees. There is a huge exotic meat trade. And I don't just mean the wet markets that you find overseas. I mean, if you go to exoticmeats.com, Salzburg, there you go, exoticmeats.com, they will sell this to you. They will ship this stuff to you. Yeah, all you have to do is cook it up. Alligator's not bad. People say it tastes like chicken, but it doesn't really taste like chicken, does it? You think so? Well, then why not just eat chicken? Why did you eat alligator? There you go. Yeah, rabbit's awesome. Ostrich, if you guys can get ostrich, pretty awesome. So there are a lot of game and wild animals out there that folks eat. And the more of it that we do eat, just the more of this stuff we kind of open ourselves up to. So kind of literally food for thought. And I see we're at about 8:15, so this is probably a good place to